unfortunately, when we wrote that down, now on the first paper we published in 2002, and we were hoping that people would read what we wrote. But pharmaceutical industry only reads what they want to read. So they read the paper and they took it. They took 5 inhibitors, so inhibitors of the same class that we had discovered. They took them to test in pain conditions. Even though we had written black on white, it is not really very good at inhibiting pain. It's good in anxiety, but not in pain.
Welcome to High Five with Dr. Igor Grant, Distinguished Professor of Psychiatry and Director of the Center for Medicinal Cannabis Research at UC San Diego. Each episode will take you on a fascinating journey into the evolving world of medicinal cannabis, as Dr. Grant poses 5 thought-provoking questions to his guest, uncovering their personal journey and unique contributions to the field. Now, here's your host, Dr. Igor Grant.
Let me introduce Dr. Daniele Piomelli, who is a good friend, colleague, supporter of CMCR for many years. Dr. Piomelli is Distinguished Professor of Anatomy, Neurobiology, Pharmaceutical Sciences, Biological Chemistry, and Louise Turner Arnold Chair in the Neurosciences at UC Irvine. Additionally, he serves as Director of UC Irvine's Multidisciplinary Center for the Study of Cannabis. Dr. Piomelli has led pioneering work into the pathways involved in the formation and deactivation of brain endocannabinoids, which we'll be discussing in a moment, uncovered important physiologic functions of these compounds, and identified pharmacological agents that interfere with their deactivation. So first of all, welcome, Daniele. Thank you so much for taking the time to join us on this podcast.
Thank you for having me.
Okay, you have done a lot of work, and we're just going to focus on one aspect of it. You've been instrumental in elucidating mechanisms of cannabinoid signaling via the endocannabinoid system, the ECS. Could you provide an overview of what is the ECS and why is it important to marijuana?
Sure. We owe this to cannabis, actually. That's one of the big discoveries that cannabis allowed us to do, because, of course, we wouldn't know about the endocannabinoid system if we didn't know about cannabis to begin with, right? And at its core, the endocannabinoid system is an endogenous signaling system, something that our body, not just our brain, our entire body, utilizes to communicate. Cells produce compounds that are similar in function, if not in structure, with the active ingredient in cannabis, which is THC, of course. And these compounds are released from one cell. They travel short distances typically, and they go into other cells where they encounter the receptors that are the same receptors that are activated by THC. They're called cannabinoid receptors, producing all sort of biological effects that depend, of course, on the cells that express that receptor and the particular state in which the cell is found. There are 2 main endocannabinoid substances. One is called anandamide. The other one has got a longer name, a chemical name, 2-arachidinoyl glycerol, call it 2-AG for short. And they're both lipids, which means they both derive from fats present in the FATs, not FACTs, fats, present on the membrane of cells. The membrane contains precursors that when need arises, so when there is a need to produce one of these endocannabinoids, these precursors are modified by enzymes present in the cells and releasing these agents. So the important thing is there are important aspects of the signaling system and make it a little bit unique compared to other signaling systems in the body. The first is that it's on demand. It's an on-demand system. So we normally have a fairly low levels of these endocannabinoids produced in a minute-by-minute way, time frame. But when we have a need, they are produced quite substantially. They act immediately, and then they are destroyed immediately. And that's the idea of an on-demand signaling. The other aspect that is also interesting is these compounds are fats, they're lipids. And that makes also a difference with respect to other transmitter systems in the body, which are mostly either biogenic amines like serotonin or amino acids like glutamate, in some cases, some gases like nitric oxide. So this is probably in the brain the most important lipid-based signaling system. But as I said, it's also present outside.
Yes. So just for, I'm sure our listeners are aware, but the transmission systems are ways of how one cell and one network communicates with another, right? By releasing a chemical messenger. And you're saying, I think, that when such chemical messengers are released, you mentioned serotonin and others, the endocannabinoids may click in. Do they have kind of a balancing action? What is it they do? Why are they released?
For a variety of different reasons, but thank you for asking that question, because it allows me to say another thing about the endocannabinoid system that I didn't mention before. So I said that it's an on-demand system produced when need arises, right? But what is the demand? Who is asking the question? And actually, who is asking the question is some other neurotransmitter. For example, glutamate, which is a very important excitatory neurotransmitter in the mammalian brain. And when glutamate is released, from the synapse, finds receptors on the cells it synapses with, and causes excitation, a variety of effects, right? In this particular case, the endocannabinoid system plays a very important role, especially in the brain, because it's activated by glutamate. There are some specific receptors called metabotropic glutamate receptors that are activated by glutamate that will stimulate the production of one of the two endocannabinoids, 2-AG. And 2-AG, what it does is fascinating because it is produced by the postsynapse. So a synapse is made up of two structures, right? One is presynapse, the other is postsynapse. So glutamate is released from the presynapse, but 2-AG is produced by the postsynapse. It's produced by the postsynapse and travels the very short space that there is between the post and the pre, and it finds cannabinoid receptors on the presynaptic bouton, is called, exactly juxtaposed to the site of 2-AG production. So the cannabinoid receptor now sees 2-AG, becomes activated, and stops the release of glutamate. So you see is a negative feedback because 2-AG is released by glutamate, which then travels back, activates CB1 receptors, which inhibit glutamate release. This is called retrograde signaling. And what makes the system even more elegant, this is already pretty elegant, but what makes it even more elegant is the fact that the destruction now, the degradation, the elimination of 2-AG, which is necessary because you cannot have a signal if you don't have something to stop the signal, that degradation occurs precisely at the presynapse. So there is an enzyme called MGL, monoglyceride lipase, which then will see 2-AG as it accumulates around the cannabinoid receptor and will cleave it into something inactive. So it's a very elegant way evolution has put on our system to modulate the effects of excitatory effects of glutamate. It is not the only thing that the endocannabinoids do in the brain. I like to say the other endocannabinoid, anandamide, does not work this way. The other endocannabinoid is also released by activation of receptors. For example, activation of the oxytocin receptor. Oxytocin is a famous peptide, is called sometimes the love hormone. It's involved in social reward. It's very important in social bonding. And oxytocin in certain areas of the brain activates a receptor that causes the release of anandamide. Now, anandamide does not travel backwards in this case. Anandamide just travels around the cell where it's produced and finds cannabinoid receptors all around and mediates the effect of oxytocin, such that if you remove anandamide, now you no longer have the effects of oxytocin. So I don't want to make this too complicated, but we basically have two modes through which the endocannabinoid system can operate. One is retrograde signaling, and that uses 2-AG, and the other one is a more of a volume transmission, so to speak, which uses predominantly anandamide, where anandamide is released and acts at a certain distance from the site of release.
So, yeah, that's very interesting. So would it be fair to say that one of the functions then is kind of a circuit breaker function? That if there is excitatory signal, you also don't want the receiving cell to get too heated up. And so the 2-AG in this case damps it down. And then it sounds like the other endocannabinoid molecule, anandamide, does more of a continued signaling function. It continues to relay a signal somewhere down the line. Is that a fair
It is.
dumbing down of, of what you've said?
Oh, it's very clear. I like the circuit breaker metaphor. It's clear. One can see it. If one measures anandamide, for example, N2G at the same time, giving a stimulation, you see that 2G goes up right away and then goes down quickly. Anandamide takes a little bit longer to rise up and then stays up and then goes down more slowly. So the two have different temporal distribution. And we don't fully understand, of course. I mean, there are many things we don't know. And this is one of them. We don't fully understand how they collaborate with one another. But there are certain conditions where they're both released, just not in the same time frame.
So can you say more about the distribution of these endocannabinoid receptors in the body as a whole and what we understand to be the main functions of that system in the body? You've mentioned the brain. What about other physiologic mechanisms here?
Yeah. So first of all, the endocannabinoid receptor, there are two of them, CB1 and CB2.
Mm-hmm.
But the CB1, which is very abundant throughout the body, is the most abundant of these receptor types. They're called G protein coupled receptors. They're receptors that have certain characteristics. They're very, very common. But the CB1 is the most abundant of them all. It's 11 times more abundant than the receptors for morphine, for example, the opioid receptor. 4 times more abundant than the receptor for dopamine. So it's a very abundant receptor in the brain. But it's also present, as you pointed out, elsewhere in the body. Actually, it's present pretty much everywhere. Every cell type we've looked in, we found some level of CB1 receptor expression. Even places where, knowing what we know about cannabis, would not have expected it. For example, in fat tissue contains CB1 receptor. Turns out that it has very, very important functions there. Liver, same thing. And then there is a CB2 receptor. A CB2 receptor is not very highly expressed in the brain, although it is to a small amount under physiological circumstances, but is expressed in immune cells outside the brain in fairly large amounts. For example, macrophage. The macrophage express it in fairly large amounts. So the macrophage are very widespread in our body. There are tissues like the skin that have up to 20% of the cell population is made up of macrophage. So clearly, in those tissues like the skin, CB2 receptors must be playing a role. We don't fully understand much about, there are some technical issues with CB2 receptors, but they clearly are spread throughout the body. So, I mean, if I were to go through a list of what these receptors do, it would take me 5 hours. But here is a very, very sort of simplistic way I look at it myself. So in the brain, to me, the most important function of the cannabinoid receptors are two. One is the circuit breaker you just mentioned before, Igor, where all sort of different type of circuits are modulated by the release of the postsynaptic release of 2AG, and that's one function. The other function, which I ascribe more to anandamide, is really the regulation of, through this sort of volume transmission, the regulation of stress coping, the ability that we have to cope with stress, which encompasses a large number of functions and processes, but it has to do with anxiety, has to do with depression, has to do even with conditions like schizophrenia. But it seems that anandamide plays an important role there. Outside the brain, I think that the most important function of the endocannabinoid system that actually brings in the brain as well is something that has to do with the fact that we need calories, we need fat to survive. So we need to look for calories, and particularly for fatty calories. We need to be able to absorb the fatty calories. We need to be able to save them in appropriate tissues like the adipose organ, and we need to be able to use them in the right time. So this series of things that are evolutionarily so important see a very, very fundamental participation of the endocannabinoid system, because one of the things that the WG mostly does in the hypothalamus, the region of the brain, is to increase our hunger for fats, our hunger in general. Another function that the endocannabinoid system has in the intestine, it facilitates the absorption of fat in the adipose organ, so white and brown adipose tissue. It facilitates the incorporation of fat into the adipocytes, the cells that make up the adipose organ. And it also regulates how fats are released, fatty acids, which are the molecular components of fat, are released into the circulation. So you can see that there is the endocannabinoid system is kind of a thrifty system, because it stimulates all this, makes us, everyone has experienced, who has tried cannabis has experienced the munchies, right? And so we know that the endocannabinoid system drives our hunger, but also it drives our ability to use whatever calories we take in through eating, and it helps store them, it helps release them. So very, very important function in evolution, and also the basis for a class of drugs, which I'm still hopeful about. They were developed in 1990. They were called CB1 antagonists, cannabinoid receptor antagonists. For example, rimonabant, a famous one, or Complia, the commercial name. They're very powerful anti-obesity drugs, because not only do they curb food intake, but they most importantly reduce the ability of fat, because they block the endocannabinoid system. They reduce the fat ability to incorporate new lipids. So that reduces the size of the adipose organ. Unfortunately, this particular class of drugs wasn't designed properly, I think, because they allowed it to get to the brain. And once you get to the brain, now you hit the problem that I mentioned before, that the endocannabinoid system is very important in regulating stress coping. So by blocking that ability to regulate stress coping, what these drugs do, they produce anxiety, they produce depression, all the way to suicidal ideation. And that was predicted based on what we know about the function of the endocannabinoid system. It didn't stop the pharmaceutical industry to move forward to a failure that unfortunately then has, cast a shadow on the field. But now, luckily, with years, there is a new hope. And it's the fact that the people have designed, very, very smart people have designed CB1 blockers, compounds that block CB1 receptor that do not enter the CNS, do not enter the brain and the spinal cord. So these compounds are excluded from the brain, so they cannot cause anxiety, they cannot cause depression.
Basically, you've also emphasized that this system has very diverse roles in the body, the brain, and one of those roles has to do with feeding behavior. And certainly by tweaking the endocannabinoid system or blocking certain receptors, you can achieve weight reduction and so forth. But then you have to look at what else is being affected by that blocking action in the brain in this case. So you're saying now there's a class of drugs that doesn't get into the brain, does not cross the blood-brain barrier, if I understand you correctly, and so may continue to have promise. Is that
That is exactly right. And, I am actually quite excited about this class of drugs. We're not involved in their discovery. It's work done by many other very good investigators. But I'm hopeful. The problem that we have is that, unfortunately, when industry gets burned on something, they tend to become like the cat who jumped on the hot stove, right? So they don't want to get even close to the stove. But, I'm hopeful that someone more visionary would be able to see this. We do have good drugs now that curb obesity, Ozempic being the classical example, but they mostly work on the intake of food. Instead, this works predominantly on the adipose tissue, so on the ability of the adipose tissue has to store new fat. Also, incidentally, fat, although it's necessary for life, I don't want to version of fat. It's a lovely, important thing. But unfortunately, under conditions of overnutrition, it will accumulate in places like the liver and producing diseases like liver steatosis that eventually can lead to fibrosis and even to cancers. So there is hope, I believe, that these agents that block the accumulation of fat, not just in the adipose tissue, but also in the liver, can be used for liver steatosis, which is a silent killer. It is, because you can't really see it until it's kind of too late, until your liver is compromised.
and steatosis is akin to cirrhosis, right? That would be
yes, absolutely.
the end stage of that, right?
Yeah, the progression is from steatosis to fibrosis, cirrhosis, and cancer eventually, liver cancer.
Well, I know that you've also been interested in developing medications that may be helpful in pain management. We know that THC, for example, does have a role in modifying certain kinds of pain, neuropathic pain as an example, which is kind of a chronic hypersensitivity pain. But you've been, I think, working on drugs that, again, may act on the degradation of the endocannabinoid system, or of the transmitters at least, that may be useful in management of pain. Can you say a little bit about that?
Yeah, no, absolutely. So the logic here is that if is involved, chemical signal is involved in producing pain relief, if we block the mechanisms that terminate the action of that signal, we should increase the effects of the signal itself. So this is not the logic that we have come up with, but it's a logic that is important. You block the degradation of an endogenous molecule, and by doing that, you boost the effects of that particular molecule. So you're not introducing a new player that, rather non-specifically, non-selectively causes, all sort of different things. You're basically asking the body to do its functions better, in a stronger way, in a more robust way, if you wish. So that was the logic behind our work on.
Could I interrupt you just for a moment to clarify? So are you saying that the endocannabinoids are involved in our body's own management of pain? And now you're talking about how do you extend the life of those things effectively by preventing them from being broken down? Is that what you're saying?
No, I was speaking more in general terms. So I was saying, so if let's say if the endocannabinoid system participates in the regulation of stress coping, so if it enhances stress coping, it helps us cope with stress. But then if we block the degradation of endocannabinoid molecules, we should get more, basically a bigger bang for our buck. The same amount. It's under the same situation, we have more endocannabinoids being produced. So we have an anti-anxiety effect, an antidepressant effect. And this has been shown in animals. I would like, maybe we can come back to that. But you asked me about pain. Pain is a different situation. And the reason why pain is a different situation, it's more complicated, is because unfortunately, whereas it's clear that the endocannabinoid system plays in the rodent, so in the experimental animals that we use in the lab, plays a clear role in pain relief, it is unclear that it does so in humans. And we know that based fundamentally on stuff that we started many years ago. Many years ago, I'm talking about 20 years ago, even more than that, we developed the first inhibitor for the enzyme that destroys anandamide, the endocannabinoid anandamide. So that inhibitor, we prepared a bunch of them, but that one in particular, if you test it in animals, it causes anandamide levels to increase. Whatever anandamide does it better. And under those circumstances, we didn't really see a whole lot of pain relief. You can tweak the system, you can tweak your experiment to get some pain relief, but you can tweak mice to do all sorts of different things. What we did see was a very profound effect on anxiety. We saw an inhibition of anxiety, an inhibition of depression. I use the term anxiety and depression. We could say anxiety-like or depression-like. These are mice and rats, so they don't have exactly the same things that we do. But all those things, all these signals that the animals are more capable of coping with stress, we could see them. We couldn't really see very much in terms of pain, at least with 5 inhibitors of this enzyme, degrading enzyme, that makes it throughout the entire body. So these are compounds that go to the brain, go to the periphery. When we tested them for pain, we didn't see a whole lot of that, a lot of analgesia, so pain relief. Unfortunately, when we wrote that down, on the first paper we published in 2002, and we were hoping that people would read what we wrote. But pharmaceutical industry only reads what they want to read. So they read the paper and they took it. They took 5 inhibitors, so inhibitors of the same class that we had discovered. They took them to test in pain conditions. Even though we had written black on white, it is not really very good at inhibiting pain. It's good in anxiety, but not in pain. And they went with that strategy and failed. and failed multiple times and kept failing. And that is a kind of a beef that I have with industry. Read the papers well. I mean, look at, don't blame animal work for being non-predictive when you're not using the prediction that the animal work is giving you. Now, it gets more complicated, and I don't want to be too technical, but if you actually make a FAAH inhibitor, an inhibitor of the same enzyme that does not enter the brain, surprisingly, this very same class of drugs, they inhibit anandamide degradation, right? But not in the brain, not in the spinal cord. When you do that, you have a much better analgesic effect. Much, much better. And there are reasons for that. The reason is that anandamide in the spinal cord does different things. It doesn't only inhibit pain, but also causes pain. It has this kind of dual action. So when you inhibit it both in the spinal cord and in the rest of the body, you're not getting the full analgesic effect. You're getting sort of an effect that is diminished by this counter effect it has. I know this is a little bit complicated, but the basic point is that if you block FAAH, this inhibitor, this enzyme throughout the body, you basically don't have a very good analgesic effect. And that's what people found in the clinical studies. If you inhibit only peripherally, you have a better effect. And now there are FAAH inhibitors that are peripherally restricted. They are being tested, not only for pain, not primarily for pain, but now primarily for chronic cough. And chronic cough is a reflex, just like pain is a reflex, but instead of causing ouch, it causes is the same thing, but with two different outcomes. And the idea there is that these compounds could be effective in the treatment of chronic cough, but they haven't been tested clinically. They have been tested the global FAAH inhibitors in pain unsuccessfully. So I would say at this point that my overall idea is that let's not waste more time and money on these inhibitors as treatment for pain, and let's look somewhere else. One thing is the other endocannabinoid, 2-HG, right? I talked about anandamide. The other endocannabinoid also has its own enzyme that degrades it. We made the first inhibitor many years ago, actually 20 years ago today, and showed that it is analgesic. Now they are much better than our own, but there are much better compounds that can be used, in the clinic. They've been tested, and there is hope that those will be good analgesics. But we don't have the clinical data for that yet.
Right. Well, but just to kind of summarize this, then, the actions of these endocannabinoids are very diverse. They are also broken down by the body. And all of these different steps offer potential pathways for therapy. But as you point out, it doesn't always work out the way one would think.
Humans are different, and we have to take that into account. But also, I think we need to be more careful, and that I say people who are in charge of discovering new drugs, more careful to the animal experiment. Just look at them a little bit more in detail, and just don't try and inject always some marketing reason to focus on a particular area. If a compound works in a certain way, respect that. Don't try and force it to do something that it doesn't do.
We need less confirmation bias and selecting the facts, I guess
We are
you'd
such
say.
biased animals, aren't we?
Let's move on a little bit if we can. So you had mentioned briefly oxytocin, which is, quote, the love hormone. What's the role of the endocannabinoid system in social interaction, bonding, these kinds of things? How would you speak to that?
That's a fascinating, understudied area of research. As you know, I think you remember from looking at our papers, I had a very brilliant MD PhD student in my lab who was fascinated with social behavior and autism. He wanted to become and has become a psychiatrist. What he found working in my lab is that basically oxytocin, which is not just a hormone, oxytocin also is a neurotransmitter. It's a peptide neurotransmitter released from the hypothalamus into everywhere in the brain. And when it's released in an area of the brain called the nucleus accumbens, which is important for reward, for our experience of rewarding events, it causes anandamide to be produced. And anandamide, as I alluded to before, activates CB1 receptors in this nucleus accumbens region. And that is critical for animals to feel the reward of being with a conspecific, an animal of the same species. Basically, anandamide triggered by oxytocin makes you like your buddy better. So strengthens social bonds and strengthens the reward that is needed to create social bonds in social animals, such as ourselves. So anandamide is actually, oxytocin has been tested for a variety of conditions, intranasal injection of oxytocin makes it to the brain. So people are testing in autism spectrum disorder, etc., with very interesting, promising results. But it is also possible that blocking the enzyme that degrades anandamide, which is produced by oxytocin, blocking that enzyme would increase anandamide when oxytocin gets activated. And so you will have a pro-social effect. So increasing the bonding, increasing the reward. And in fact, people have been testing this. Recently, there was a clinical trial with a FAAH inhibitor, so an enzyme, the FAAH is the enzyme that destroys anandamide, a FAAH inhibitor in autism spectrum disorder with very promising results. And this is a phase 2 trial, which is not conclusive, but it's an efficacy trial. So the results were clearly indicating that blocking FAAH in the CNS, which this compound is a Johnson & Johnson compound, does, blocking FAAH in the CNS has positive effects in autism spectrum disorders. And autism spectrum disorders are a social problem. It's a problem with sociality. People who live with autism spectrum disorders have a difficulty in understanding others as being part of the same social group. There is a lack of what is called a theory of mind, basically, the idea that others can have thoughts, right? Like I do, right? I mean, I'm looking at you and I know that you're pondering what I'm saying. So I know that you have a mind. And that is a problem for an autistic person who does not have, in many cases, that ability. And so I'm hopeful that FAAH inhibitors will find use for that condition. The other condition in which they seem to be fairly effective is anxiety. There are some suggestions, again, a lot of preclinical work, but also some clinical studies with positive results, although not definitive results on anxiety. And if you think about it, two things are connected, right? Because I think they're connected, especially in social anxiety, because our relation to the world and our relation to others is the fact that anandamide, the endocannabinoid system, is involved in both of them, makes a lot of logical sense.
So without joking too much, why, as a child of the 60s, I remember hippies walking around saying, make love, not war. And a lot of them were, of course, smoking marijuana. So do you think there's something to that? And can the exogenous cannabinoids also be helpful in this area, do you think?
Well, I think there is something to that. I was not a hippie. I never really, that's not where my ideas are. But I do know
We need to socialize you more, I guess, Daniele, right?
I'm kind of a bear that way, but I like social. I like people.
Yeah, I know. I'm joking.
I was quoting George Carlin. Anyway, the comedian George Carlin. I think you hit a very interesting point there with interesting, socio-political ramifications. I believe that one of the reasons why the war on drugs started in the 1970s, with the Controlled Substance Act that is still, uh, with us and is still a major barrier to productive research in the cannabis space. The reason why cannabis was delegalized in the 1970s with the Controlled Substance Act was in large part because the college youth, was seen who was using cannabis and wanted peace. That remember, there was during the Vietnam War, the disastrous war where hundreds of thousands of Americans and millions of Vietnamese died for no reason. It was seen as a threat to the cohesiveness of the pro-war efforts. And so you're definitely right. If you activate the endocannabinoid system, you don't want to go to war. Depending on the situation, depending on the personality, and depending on the ideology, this could be a bad thing or a good thing. To me, is in general a good thing, but except very rare exceptions.
Great. Well, our time is going to come to a close. And I guess my last question might be, you've done extensive research in this area. Is there something that surprised you along the way, or even putting it a different way, has opened up kind of your eyes to new opportunities? So either surprising or kind of
It's an interesting question. I got to say, first of all, I consider myself one of the luckiest person on earth because, I bumped into the endocannabinoid system when it wasn't a system, didn't have a name. This was back in the 1990s. And I've been surprised basically every other day. Every new discovery was in part a surprise. Not all of them, but many of them were really weird, stuff that I did not expect. But there is one maybe that I could single out because it's something now we are really excited about and working on, is the fact that we discovered actually early on, literally 40 years ago, that the endocannabinoids don't work alone. They don't work alone in the sense that, I said, they're released upon demand, they act in a variety of different ways, and you would think, okay, that's it. No, in fact, what happens is that they are released at the same time with other compounds that act as ancillary, have an ancillary function. And these compounds can either be synergistic, which means that they potentiate the effects of the endocannabinoids, or they can be antagonistic, which means that they block the effects of endocannabinoids. One of these compounds that I've been working on really since inception is similar in structure to anandamide, but dissimilar enough that it does not bind to the cannabinoid receptor, but similar enough that is produced through the same pathways and can be cleaved, destroyed by many of the same enzymes, so many of the same mechanisms. And I think now that the future, one of the important set of discoveries for the future will be to understand the function of these compounds, which we call para-cannabinoids. P-A-R-A, cannabinoids like paramedic or paraphrases. Para means something around. It's a Greek word, para, which means something around. So they are around the endocannabinoids. And they don't necessarily do the same things that they do. They do sometimes the opposite, sometimes they potentiate, but they work in concert with the endocannabinoids. So, for example, in the case of pain, especially in the periphery of the body, anandamide is released along with PEA, with palmitoylethanolamide, this para-cannabinoid. And the two of them, when they act together, if anandamide produces 2 and PEA produces 2 when they are given alone, an effect of equal 2, if you put them together, the effect is 10. You have what is called a synergism with potentiation. So there is an element of a mysterious element there. The body clearly has achieved a way of obtaining maximal effects, combining different chemicals. And I would like to pursue this. I hope other people will be interested in pursuing it. And I hope, given the circumstances, we will still have the possibility of pursuing them scientifically with funding from the federal government. But that's a matter for a different webinar.
Yeah, maybe we need more pro-social medications in some aspects of the federal government.
Now, here is an idea. We
won't go there.
We could rent a plane and spray anandamide all over the White House and see what happens. Maybe someone will get nicer.
Well, Daniele, thank you so much. This is a very complex area, and I think you have explained some things that textbooks are written about in a way that I think our listeners can appreciate.
Thank you for having me.
Thank you very much for taking the time.
My pleasure.
Please continue your wonderful work, and I look forward to continuing our collaborations and discussions.
I'm always very grateful to you and the CMCR for all the work you guys have done over the last, 25 years. And I'm looking forward to your next meeting and new stuff coming from your wonderful work. Thank you again for having me.
Thank you very much. Bye-bye.
If you like this podcast, please subscribe so you don't miss future episodes. For more information on the Center for Medicinal Cannabis Research at UC San Diego, please find us on social media at CMCR_UCSD on Twitter or X, Instagram, and other social media platforms. Or visit our website at cmcr.ucsd.edu. Thanks for listening.